Hello and welcome. Today, we're diving deep into a bacterium that's really starting to make a name for itself in clinical microbiology, Peptostreptococcus anaerobius. We're going to explore why this isn't just another gram-positive coccus you can ignore, and why getting the ID right down to the species level is becoming so incredibly important for our patients. So let's kick things off with the big question, right? The one that really gets to the heart of this whole discussion. Why can't we just report gram-positive anaerobic coccus and call it a day? Why isn't that good enough anymore? Well, as you're about to see, the answer to that question changes everything when it comes to treatment. All right, here's our game plan. We're going to follow the story of Peptostreptococcus anaerobius, starting with its life as a quiet member of our microbiome, and then see what happens when it turns into a serious pathogen. We'll start in the lab, looking at how to nail the identification. Then we'll jump over to the clinical side to see the diseases it causes, including a pretty shocking link to cancer. And finally, we'll get into the real nitty-gritty, treatment guidelines and the massive challenges we're all facing with antibiotic susceptibility. Okay, first up, an overlooked, opportunistic pathogen. Let's really get to know this organism because it truly lives this fascinating double life. So what is it exactly? Well, the textbook definition is pretty straightforward. It's an obligate anaerobe, meaning it hates oxygen. It's a gram-positive coccus, and it doesn't form spores. But the key thing to remember is where it lives. This bug is a totally normal, everyday resident of our gut and the female genitourinary tract. When everything's fine, it's just there, a quiet neighbor. But here's where the story takes a turn. That quiet neighbor can become a real problem when our body's defenses are down. Think about it. Trauma, a major surgery, or even a growing tumor can break down the physical barriers that keep it in its place. And when that happens, this harmless commensal transforms into a pretty formidable opportunistic pathogen. And that means, for all intents and purposes, these infections almost always come from inside us. All right, let's head over to the lab. How do we actually figure out that this is the bug we're dealing with? This is where being a good detective really pays off, because there are a few classic clues that can point you in the right direction. So the classic workup, right? It starts with the gram stain, and what you'll see are your typical gram-positive cocci, usually in pairs or short chains. On an anaerobic plate, it's actually a pretty fast grower for an anaerobe. You'll get these gray, shiny colonies within about 48 hours. But the real dead giveaways? First, there's this weirdly distinctive, sickly sweet smell. And second, its unique susceptibility to SPS, sodium polyenethyl sulfonate. If you see a zone of inhibition between 12 and 18 millimeters around that SPS disc, you should definitely be thinking Peptostreptococcus anaerobius. Now this is a really cool and practical way to tell it apart from its closest cousin, Peptostreptococcus stomatis. Honestly, the specimen source is one of your best clues. Just think. Peptostreptococcus anaerobius is your below-the-waistline bug from the GI tract or a GYN source. Peptostreptococcus stomatis, on the other hand, is from above the waistline, usually the mouth. When you combine that with the SPS zone, see how P. stomatis is way more susceptible with a much bigger zone? You can get to a really confident presumptive ID right at the bench. But of course, we have to talk about the real game changer in the modern lab, and that's MALDI-TOF. I mean, this technology has totally revolutionized anaerobic bacteriology, hasn't it? It gives us a fast, super accurate, and cheap species-level identification in just a few minutes, straight from the colony. It's without a doubt the gold standard now for routine ID. Okay, so we've identified it. Great. But why do we care so much? Why was all that work important? Let's take a look at the seriously broad spectrum of diseases this organism gets involved in. Peptostreptococcus anaerobius is a major player in a whole host of really nasty polymicrobial infections. We're talking intra-abdominal abscesses, pelvic inflammatory disease, those awful infected diabetic foot ulcers, and even life-threatening stuff like necrotizing fasciitis and brain abscesses. It's everywhere. But it's not just a sidekick in a mixed infection, no. There's a much more complex and, frankly, more sinister story that's starting to come to light. Yeah, you read that right. Recent research has uncovered this shocking connection between Peptostreptococcus anaerobius and colorectal cancer. It's not just hanging around. It seems to be actively colonizing tumor tissue and acting as an oncombrobe. Think about that for a second. That means it might be promoting tumor growth and, what's really terrifying, making cancer cells resistant to chemotherapy. This discovery just completely changes how we have to think about this bacterium. It's not just an opportunist anymore. It might actually be a driver of cancer. So that brings us right to treatment. I mean, given how serious these infections can be, how do we actually manage them effectively? Okay, let's take a quick look at the Dutch swab guidelines for adults. You can see a pretty clear pattern here. For something really severe like necrotizing fasciitis, they recommend a heavy hitter like meropenem, often with clindamycin thrown in to help shut down toxin production. For pelvic infections, you'll notice metronidazole is a real cornerstone of therapy because it specifically targets the anaerobes in these mixed infections. And if we look at international guidelines, like from the IDSA, they're saying pretty much the same thing. The number one takeaway is that your empiric therapy absolutely has to provide solid anaerobic coverage. So drugs like piperacillin tazobactam or the carbapenems are your go-to choices. But, and this is so important, none of the antibiotics matter without aggressive source control. You have to drain the pus, you have to cut out the dead tissue. That part is totally non-negotiable. And now we've arrived at what might be the most critical and honestly frustrating part of this whole story, especially for us in the lab, the antimicrobial susceptibility challenge. It's a big one. We're pretty much flying blind, interpreting results in a data vacuum. And here is the bombshell. As of early 2024, EUCAST has not published any official clinical breakpoints for Peptostreptococcus anaerobius. Let that sink in. This is a huge gap. It means we, in the lab, cannot officially report an isolate as susceptible or resistant using a standard, validated framework. So what does that mean for our day-to-day work? Well, it means we can't just run an AST panel and spit out an S or an R. Instead, we have to report the raw MIC value and then help guide the clinicians by looking at published data on MIC distributions and offering our expert interpretation. Our role as consultants becomes more important than ever. So this is the kind of data we have to lean on. Just a quick refresher, the MIC90 is the concentration of antibiotic that stops 90% of the isolates tested. So take a look at penicillin. The MIC90 is 32. That's clearly in the resistant range. It tells us that while some strains might be susceptible, a lot of them are not. But then look at metronidazole and imiquimod. They look great, consistently active. This table is basically our best tool for interpreting a raw MIC value for a clinical isolate. And this chart just drives the point home, especially for clindamycin. Some studies are showing resistance rates of over 50%. Think about that. Using clindamycin empirically for an infection where you suspect this bug is involved, it's literally a coin toss whether it's going to work or not. That is critical information that our clinical colleagues need to have. Okay, let's start to bring this all together. What are the big picture takeaways for preventing and controlling these infections in your practice? Well, since these are endogenous infections, they come from within, prevention is really all about keeping the organism from getting into places it shouldn't be. That means super strict aseptic technique during surgery and making sure you're using the right perioperative antibiotics that actually cover anaerobes. For us in the lab, it's a biosafety level 2 organism, so standard precautions apply. And the good news is it's easily killed by our standard hospital disinfectants. We have definitely covered a ton of ground in this explainer, and all of this information was pulled together from a wide range of sources. If you're a real keener and you want to dive into the primary literature yourself, we've got you covered. A full list of all the references is available for you in the accompanying notebook document. So, we'll finish with a question for you. What are the biggest headaches your lab runs into when you're dealing with anaerobes like Peptostreptococcus anaerobius? Is it the lack of breakpoints, the quality of the specimens you get, or something else entirely? Let us know your experience down in the comments. We'd love to hear it. And hey, if you found this helpful, make sure you subscribe for more deep dives like this one. Thanks for tuning in.